Good afternoon, all of you. Welcome to online lecture series. Myself, Mrs. Ashwini Kadbhane from Department of Microbiology, S.V.K.T College, Devlali Camp. Today we are going to see TY-BSc paper number 3, that is the metabolism. In our previous lecture, we have seen about the active and the passive transport. In active transport, we have seen the primary and secondary. Today we are going to learn about the third type, that is the endocytosis. Okay. So in continuation with that, you have to see the previous lecture for the continuation purpose. So this topic is for the TY-BSc students of the semester number 6, paper number 3, that is the metabolism. So what we have learned earlier, that we are going to see. So let's recap what we have seen earlier. We have seen earlier the active transport mechanism, we have seen its types, we have seen the primary and the secondary mechanism. In primary, we have seen the ATP derived pumps. ATP derived pumps means the metabolism or the permeability of the cell membrane by use of the ATP energy. Okay, in that we have seen the various pump, that is the F, V and the P type of the pump. The P type, on the name it is the phosphorylated pump, in which the ATP is going to be phosphorylated. In F type of pump, the ATP is going to be phosphorylated also and synthesized also. And in the V type of the pump, again the ATP would be synthesized. So that we have already learned in our previous lecture. Next we have seen about the ABC transporter, in which the various carrier proteins they are involved, the various channels they are involved for the transportation of various nutrients in and out of the cell. Next we have seen the secondary active transport. In that we have seen the same port and the antiport. Same port means two molecules moving in the same direction, that is from the low to high, high to the low molecular, and in antiport two molecules they are moving in the opposite direction, but main gradient, that is the high to low, but only they are moving in the opposite direction. So this we have already learned. So today our agenda of this session, of this lecture, is to learn about the third type of active transport, that is the endocytosis, in which there is also the movement of molecules from the high to low, but how they are moving, this type of mechanism, it is generally done in the vesicle. Okay, so it is the endocytosis. So in endocytosis, we are going to learn the three types, that is the phagocytosis, the pinocytosis and the receptor mediated cytosis. And the last we are going to learn the exocytosis also. So today we are going to learn about the third type of active transport mechanism, that is the cytosis. Before that, we are going to see what is active transport. To move the substance against a concentration or the electrochemical gradient of the cell, energy is required, because here we are moving the molecules from the against the concentration gradient, that is from the high concentration towards the low concentration. In fact, cell spends much of its energy for the metabolism purpose and for the active transport process. Most of the RBC, they are using their internal energy for the maintenance of internal sodium and potassium level in the RBCs. And so this, it is the against the concentration gradient. Here most of the ATP is being hydrolyzed or being phosphorylated. So such type of transport mechanism is also called as the uphill transport mechanism. So we are going to see one diagrammatic representation of the active transport, which we have learned earlier, and now what we are going to learn. So in active transport, cell transport, the two types, that is the passive and the active. In passive, we have already seen the simple diffusion, the osmosis and the facilitated diffusion. In that, some require a carrier protein, some require a channel. Osmosis, it requires the aquaporins. We have already seen where the regulation or the transportation of water molecules is going to be held. In simple diffusion, the non-polar molecules and the small molecules, they are going to be transported. Okay, in facilitated, the polar molecules, the charged molecules, they are going to be transported. So that we have seen how do they transport, what are the various mechanisms. In active transport, we have seen the two types, that is this primary and the secondary. Both comes under the pumps, that is they require the energy. Okay, they require energy. So it is being classified under the category pump. Today's our lecture, it is been in the vesicle. Okay, here the vesicle they are involved, in that the exo and endocytosis occurs. So both type we are going to see. In endocytosis, the three types, the pinocytosis, the phagocytosis and the receptor mediated cytosis we are going to see. Phagocytosis already you have learned in your immunology, that is the engulfment of the foreign material or the eating phagosome. They are the eating molecules, eating the antigen or eating the foreign material which has been incorporated in your body. So that also we are going to learn in the active type of transport in the vesicle. So before starting our lecture, we are going to focus on the endocytosis. So endocytosis, it is a cellular process in which substances are brought into the cell. Endo means inside. Cytosis means killing. So in the name itself, it is giving us the definition of the endocytosis. Endo means inside the cell, the molecules, they are being trapped and then they are being dissolved in the cell membrane. Here there is no killing, because here we are learning this under the active transport mechanism. Okay, so here the dissolvement of the molecules is been there. So endocytosis, it is a cellular process in which substances is brought into the cell. The material is to be internalized, is surrounded by an area of a cell membrane which then bursts off inside the cell from the vesicle containing the ingested material. Means once the material go inside, penetrate inside, they bulges out. There is one bulge comes out, okay, and that bulge, it contains the ingested material. So endocytosis includes the two types, mainly three types, that is the pino, phago and the receptor mediated. So endocytosis, again, it is the active process of taking in something through a cell membrane which also require energy, which is ATP is involved. In this case, when a molecule causes the cell membrane to bulge inward, it is the endocytosis, forming a vesicle. Here the inward bulges form. So here we can see the diagram, in the diagrammatic way, the three types of the endocytosis. Okay, first you can see the phagocytosis. In first figure, in phagocytosis, you can see a solid particle. It is being trapped by the pseudopodium. The plasma membrane contains a pseudopodium. Pseudopodium means it is the false lag. Okay, in that false lag or in that pseudopodium, the solid particles, they are being trapped. As they are trapped, they are bulging inside, as it is the endocytosis type. In exocytosis, the bulge would be in the outer side, that is it would be in extracellular fluid. Here the bulge, it would be the inside the plasma membrane. That bulge goes inside the phagosome, makes a food vacuole inside that. That food vacuole, that particle, then it is liberated inside the plasma membrane, and such type of endocytosis occurs. In pinocytosis, you can see the two bulges, they are being formed, and the bulge, it becomes a vesicle. In phagocytosis, it is the phagosome, and here they are the vesicle. Okay, the vesicle is formed, and that vesicle, they liberate the particle or they liberate the molecule inside the cytoplasm. In the receptor mediated endocytosis, you can see there are the molecules, that is the receptors, they are present. And on that particular, as compared to enzyme and substrate, particular enzyme required a particular type of substrate. Same here, a receptor required a particular type of substrate, and only that molecule would be transported inside the cell membrane. Here the coated pits are there. On that coated pits, extracellularly there is the receptor. The receptor molecules, they are attached to the particular substrate. Okay, so only that substrate would be come inside the plasma membrane, and there is the coated vesicle formed by that substrate, which includes the outer membrane. There is the coat protein, and inside that there are the receptor and the molecules, and then they are liberated inside the cytoplasm. So these are the three types of the endocytosis in the diagrammatic way. We are going to learn one by one each and every type and how it is being liberated or how it is being transported from the higher concentration towards the lower concentration. Okay. So moving towards first endocytosis, in which the molecules they are being trapped, the bulge would be formed inward in the interior part, and that interior part would form a vesicle which also includes the molecules, and then they were the liberated. So this is the endocytosis. Endocytosis, they require the ATP. Okay, these also, as compared with the primary and secondary, these also includes the ATP, because this is comes under the active transport, and active transport itself, it is the ATP hydrolysis and ATP phosphorylation. Because of that, ATP energy is given out, and that is utilized by these types of transport mechanisms. So pinocytosis, it is usually occurs from the highly ruffled regions of the plasma membrane as the invasion of the cell membrane to form a pocket that we call a bulge, which then pinches off into the cell from a vesicle, which is 5 to 5 μm in the diameter, filled with a large volume of the extracellular fluids and molecules within it. The filling pockets occurs in a non-specific manner. The vesicle then travels to the cytosol and fuses with the other vesicles such as the endosomes and the lysosomes. So pinocytosis. As in the phagocytosis, a vesicle is formed which contains the molecules that are brought throughout the cell, into the cell. Here vacuoles and vesicles, they are involved, produced by the phagocytosis and the pinocytosis. They can fuse with the lysosomes. Lysosomes, they are the vesicle, they are the digestive enzymes which digest the material which is brought inside the cell by the vesicle. You can see in the diagram that there is the bulge, and that bulge comes inside the cell in the cytosol, and that we call as the vesicle. That vesicles contains the lysosome, which is the enzyme, digestive enzymes which degrade that molecules. Phagocytosis and the pinocytosis remove the membrane from the cell surface from the vacuoles that contain the engulfed material, that is the cell drinking. It is also called as pinocytosis, also called as cell drinking. Pinocytosis is referred as engulfment macromolecules. Phagocytosis is referred as engulfment eating cells but micromolecules. Pinocytosis macromolecules occurs when the external fluid is engulfed. Okay, when the external fluid is engulfed, at that time pinocytosis occurs, which involve the eating of the macromolecules of the cell. You can see in the diagram pinocytosis. It is a type of endocytosis, a plasma membrane. The molecules, they are being attached on the membrane. Okay. Then they are being penetrated inside. Here the fluid is also penetrated. You can see the fluid also coming with the molecules. The bulge is formed, the vesicle is being bought, and the lysosome enzyme digests that molecules inside the cytoplasm. So this is the pinocytosis. In this process, the pinocytosis, the plasma membrane forms an invasion. Whatever substance is found within the area of invasion is brought inside the cell. Whatever, it may be a liquid or it may be a solid. Both types of molecules, they are being trapped and brought inside the cell by the pinocytosis mechanism. In general, this material will be dissolved in water, and thus this process is referred as cellular drinking. Why? Because it also includes the water molecule. Here not only the solid material, not only the molecular substances, but also the water molecule being trapped, and they are being utilized by the cell. So it is also called as the cell drinking, to indicate the liquids and material dissolved in the liquid is ingested by the cell. This is opposed to the ingestion of the large particulate material like the bacteria and other cells or the cell debris. You can see in the diagram the three types. Again, I have taken one diagram which will show a clear picture of these three types. In phagocytosis, you can see only the molecules, they are being trapped, a bulge that is formed, then the phagosome taking that molecule inside the cell that contain a lysozyme enzyme which then will degrade that molecule or eats that molecule. Okay, and then it will incorporate in the cytoplasm. In pinocytosis, you can see the movement of molecules with the liquid. There is the liquid also being trapped by the pinocytosis, because it is called as the cell drinking. Cell drinking means here the involvement of liquid is also there. So this would be brought inside by the vesicle, and the vesicle will contain the lysozyme enzymes. Again, they would be degrade that material, so it is called as the pinocytosis. And the third, you can see the receptor mediated cytosis, in which the molecules, they are being trapped by a receptor. Specific molecules, they are only trapped. A bulge is formed, and then same process, the vesicle is formed, and the molecules, they are broken down or they are utilized in the cytoplasm. Here you can see in the third part, the receptor is used. So it is called as the receptor mediated endocytosis. In previous two, phago and pinocytosis, no receptor is mentioned over there. So any molecule can be come inside. Here in receptor mediated, only specific molecules comes inside the cell, and they are being eaten or being transferred into the cytoplasm. So again we have seen about the three types. In next slide we are going to see the in detail manner. Before that, what is exocytosis, that is also we are going to learn. Exocytosis is exact opposite to the endocytosis, but it is active transport. Here the bulge would be found on the outer side. Okay, it is the movement of moves the material to the outside from the cytoplasm towards. Here also the concentration gradient, it is against the concentration gradient, but some molecules, they are moving from the cell toward outside. So that process is done by the exocytosis. A vesicle fuses with the plasma membrane and discharge the content outside. This allows the secret molecules the incorporation of the materials from outside the cell by the formation of vesicle in the plasma membrane. The vesicle surrounds the material so the cell can engulf it. The fusion of the vesicle to the plasma membranes adds the membrane to the cell surface. Here you can see again the exocytosis, in which a cytosol the transport vesicle, it attached to the plasma membrane. You can see in the diagram, it is being attached to the plasma membrane, then the plasma membrane get opens, and due to the opening of the plasma membrane, the molecules, they are being liberated outside the cell, that is the extracellular, in the extracellular part. Outside the cell, the proteins or any molecules which are being transported, they are being transported outside the cell by again the cytosol and the transport vesicle. Okay. Exocytosis, it is the fusion of secretory vesicles with the plasma membrane and result in the discharge of vesicle content into the extracellular space and the incorporation of new protein lipids into the plasma membrane. So you can see again in the diagram, first it have the cell membrane. First step, the molecules, they are being trapped in the cytosol. Then the molecules, they are being transported to the vesicle. The vesicle is attached to the plasma membrane. The plasma membrane gets open, then the molecules, they are being liberated in the outside or the extracellular part of the cell. Again you can see in the diagram, vesicle, second is the extracellular fluid, and third it is the proteins. For example, we are taking the proteins, they are being transported in the stepwise manners. So this is the exocytosis. Now you can see in the diagram the endo and exocytosis distinguishing characters. So in first, you can see the endocytosis, the molecules being attached to the plasma membrane. Then there is the bulging out of the molecules. The bulge form, the bulge, it is trapped inside by the phagosome or the vesicle. And there are the food digestive enzymes, example lysosome. That lysosome that digests the whatever that is come inside the cell. Then the digestion takes place. The molecules which are being digested, they are being utilized by the cell. Whatever it is remaining, or when it is the high concentration that is moving towards the low concentration, then the lysosome fuse with the secondary part. The cytosol, it is formed by the cytosol. It is form of vesicle. That vesicle, it is again attached to the plasma membrane. The plasma membrane gets open, and the food particles is thrown out of the cell by the exocytosis. So this is the process, that is the endo and the exocytosis. Okay. Now the next part of the endocytosis, it is the phagocytosis. A vacuole is formed containing the material that is being engulfed. which is known as cell eating. Pino is known as the cell drinking, and phagocytosis is known as the cell eating. Phagocytosis is the cellular process of engulfing of solid particles by the membrane. Here the only solid particles they are being engulfed, and in pinocytosis the water and the solid particles, okay, to form an internal phagosome or the food vacuole. The phagosome is usually delivered to the lysosome, an organelle involved in the breakdown of the cellular components which fuses with the phagosome. The contents are subsequently degraded and either released extracellularly via exocytosis or released intracellularly to undergo the further processing. Phagocytosis involves the acquisition of the nutrients for some cells, and the immune system is the major mechanism to remove the pathogens and the cell debris of the phagocytosis. Phagocytosis is involved only in some part of the active transport for the nutrient type, but mostly the major part of the phagocytosis, it is utilized in the second line of defense mechanism where the pathogens or the foreign material, they are being trapped, they are being eaten, and they are being thrown out of the cell. Bacteria, dead cell tissue, and the small mineral particles, they are the examples of object they are being phagocytosized. Phagocytosis is the specific form of the endocytosis involving the vascular internalization of the solid particles such as bacteria. Next, it is the receptor mediated cytosis, in which already we have seen one receptor is needed. A receptor helps to attach the molecules to the membrane before taking them in. Macromolecules binds to the receptor on the surface of the cell. A receptor with bound macromolecules aggregate in one area and brought into the cell by the endocytosis. The vesicle containing the macromolecules can release the macromolecules into the cell directly, or they can be processed by the chemicals contained with the lysosome or the fusing of the lysosomes. Vesicle and the receptor then returns to the cell surface. Receptor mediated endocytosis occurs when the material to be transported binds to the uncertain or the certain specific molecules within the membrane. Example of the receptor mediated is the transport of insulin and cholesterol in the animal cell. In our cell, the transportation of the insulin and the cholesterol in and out is done by the receptor mediated endocytosis, whereas receptor is needed for the binding of a macromolecule. Okay. So in the diagram, you can see the receptor mediated endocytosis. You can see the example, it is the calretitin. Here is the receptor protein, coated protein, target molecules, they are being attached to the receptor protein. Then there is the coated vesicle, and the vesicle, it is being bulges inside the intercellular and it is being taken in the cytosol. And there the lysozyme enzyme or the fusion of lysozyme enzymes comes and they degrade that molecule. So here we have completed our endocytosis and exocytosis. What we have learned, we are going to see in a summary. So here in active transport, we have seen it is the type of the transport mechanism where the involvement of energy or the ATP is there. We have seen its types, that is the primary and the secondary, in our previous lecture. But today we have seen the third type, that is the endocytosis, which involves the pinocytosis, that is the drinking of the cell, phagocytosis, that is the eating of macromolecules, and the receptor mediated cells through which insulin and the cholesterol be transported in the animal cell by using such type of mechanisms. And last, it is we have seen the exocytosis, which is also a type of the cytosis in which the exact opposite mechanism of the endocytosis being occurred. So this is our summary of today's lecture. So all these points we have seen in our today's lecture. So today we have completed our active transport mechanism. In our next session or in our next lecture, we are going to see the third type, that is the group translocation of sugars in the bacteria. Okay. We have seen glucose that contain the GLUT1, GLUT2, GLUT3, all the type of carrier, and through which the glucose is being transported. But what about other sugar? How they are transported? How they are incorporated in the bacterial cell? That we are going to see in our tomorrow's lecture. In that we are going to see the PEP transfer, that it is the pyruvate enol phosphopyruvate enol transferase, okay, phosphotransferase, in which the release of energy from PEP molecule is there, and that energy it is being utilized by the various enzymes for the location of the sugar. In that, the sugar is not only localized but it is also transformed. Means, for example, glucose is transformed into glucose 6-phosphate. Okay. In group translocation, the sugars, they are not only being transferred, not only being translocate, but also being transformed. That we are going to see in our next session. And the next part, that is the ionophores, why they are utilized, what are their mechanisms, and what are the examples, we are going to see in our upcoming lectures. Okay. Thank you for attending. Have a good day.